the talk by Jennifer Graham, who's working with the USGS out of Troy, New York. And she's going to be telling us what's going on with the USGS and cyanobacteria. Is that correct?
Yes.
Okay.
Yeah, just some of the monitoring strategies that are being used across the nation.
Thank you.
So we talked about it this morning, but when we're talking about monitoring for cyanobacteria, this is always the challenge that we're dealing with. These things move, and depending on when you're there, you can have a very different picture of what's happening in the environment. This is an example from 2006 in Iowa when we were just figuring out what was happening with these things, and the DNR saw this sample and said, we're closing the beach. They didn't even test it. They said, we're done. The news reporters came out a couple of days later, and the beach was crystal clear. So then you had all the news reports that came out and said, this is, you know. So understanding this phenomenon is something that we always have to take into account when we're developing our monitoring strategies. And we need to think about why we're monitoring, whether it's for recreational activities, drinking water, or for research. And the approaches that we use are going to be different, and the tools that we use are going to be different. So what I'm going to do today is give you a very high-level, quick overview of a lot of the tools that are in use. And these are all tools that the U.S. Geological Survey is using and exploring. A lot of the work that we do is in collaboration with partners at local, state, and federal level. So these are all things that are being used and explored. And one of the points that I really wanted to emphasize is that citizen science is becoming increasingly important when we're talking about blooms for the reason that I just mentioned. These things are temporally variable. Things like bloom watch are so important to capturing that variability and knowing that the samples we collect at a given point in time may not always be the best representation of what's going on. The other thing to keep in mind when we talk about cyanobacteria blooms is that this is a cellular phenomenon, right? These are microorganisms, and they're manifesting in ways we can see with the naked eye and we can see from space. So there are a lot of different kinds of tools that we can bring to bear. And like I said, this is just going to be a very high-level overview. I could probably talk for 10 minutes about any single one of these tools that I'm going to touch on. The first is the screening tools, and one of my favorites, it continues to be my favorites, is if you have nothing else, if you can see it or if you can retain it on a large mesh net. Nancy mentioned the 50 microns. In my research, we've used 100 microns. If you can see it, you probably have something there. You might have toxin, you might have taste and odor. Some of the work I did with Springfield City Utilities in Springfield, Missouri, their limnologist said, I can predict 2 weeks in advance when we're gonna have a taste and odor event because I'm retaining organisms in my net. We don't pick them up in our traditional counts, but I see them in my net and I know it's coming. And that rule of thumb has really stood the test of time. So again, if you have nothing else, a net, something that you can retain on a screen, is a good indicator that you have a potential for toxins there. And that's a tool that's very widely used and very easy to use. Another one that's very easy and we're seeing used more and more often is the use of cameras, whether as like bloom watch, you're using cameras in your cell phones, cameras mounted on airplanes to look at aerial extent, or time-lapse cameras that are again capturing the variability. This is from a study that USGS did in Kansas where we were looking at recreational areas. We set cameras up to look at different areas to take pictures. Initially it was hourly, and we looked and we said that's not good enough, and eventually we had dialed back till we were taking pictures every 5 minutes. Here's just an example of a 15-minute offset, and this kind of information is really useful, especially when communicating to the general public about why things might be considered a potential risk even if you can't see it when you happen to be there. Test strips are used, they're used when information on toxicity or potential toxin presence is needed. These tools are considered rapid screenings, but they take 15 minutes to an hour, and there have been mixed results on how these are used. The interpretation is difficult and highly subjective. They work better when you have higher cyanotoxin concentrations. And I think it depends on the organisms that you have present in your sample too. I showed this slide in a different talk, and somebody in the audience says, I use them all the time and they're great. I think they're great. And I think that really is because the system that he is using, for whatever reason, this approach works well. But it is something that you can get a rapid plus minus result fairly quickly. Again, it's an hour, so there's still some time there, but it's out there and available. Genetic tools are becoming increasingly common. They give us a lot of information on whether the potential for cyanotoxin production is at a site or not. It's a screening that way. They can't necessarily tell us whether toxins are there or not, but they can tell us whether the potential for production is there. And these types of tools are being incorporated into screening programs. The Ohio EPA has adopted some genetic tools into their protocols. And there's some evidence that at some sites, at some times, the genetics may give an indicator, an early indicator that toxin production may be occurring. In this example, there's a couple weeks offset between cyanotoxin detection and detection of the genes in the environment. So again, I think this is a running theme, is not all approaches work in all systems at all times, but there are a lot of tools for us to use and explore and evaluate what works best for our needs in the systems that we're studying. A lot of work that we've done in the USGS is with fluorescence sensors. I know the Monitoring Collaborative is also using the fluorescence sensors. A lot of our work is with the inline continuous fluorescence sensors, and learning how to interpret those data were a challenge, but there are patterns there that we can use to anticipate whether or not we have a bloom present. All of these examples that I show here have really noisy patterns in signal. And if we're thinking of traditional water quality variables like temperature or dissolved oxygen, you might look at these data and say, these are no good. There's something wrong with the sensor. There's something happening here. And when we started really interpreting these data and looking at what's going on in time, these are blooms, and it's indicative of that rapidly changing condition. And this, just pointing at this example here from Kansas, we can tell when the cyanobacteria bloom really initiated, because we went from very little noise in our data stream to a whole lot of noise. So again, these are very useful and helpful in the environment. We can also use those fluorometers to develop models to estimate cyanotoxin concentrations or the probability of cyanotoxin occurrence in real time. And we've had success with this at several sites throughout the nation. And again, it depends on the system and what's there. Satellite imagery is becoming increasingly common. It allows you to look at different scales. Spatially, you can look across a large region. You can also look forward and backwards in time. I think that the satellite technology is emerging. This is an example from the Cyanobacteria Assessment Network project, which really can't resolve most of the small lakes that we've been talking about here today. But there are satellites available with better spatial resolution that are being explored to evaluate cyanotoxin. And then finally, I wanted to just touch on some of the new and emerging technologies that are out there that allow assessment at new spatial and temporal scales. There's passive samplers that we can deploy in the environment that can monitor for us when we're not there. There's work going on with characterizing the hyperspectral signatures of cyanobacteria, so we may be able to look at them with non-contact sensors, or flying over a site or an area with planes with hyperspectral sensors mounted on them. There's imaging flow cytometry, which allows a look at algal community assemblages. There's environmental sample processors that are out there collecting samples, analyzing them in real time. They're kind of like this mini labs out in the environment. And there's a host of new types of sensors, more complex fluorometers that are available for use. A lot of these are still under research and development. A lot of these are still very expensive and cost prohibitive for most programs, but I'm optimistic that we're going to start to see some of these become more reasonably priced as, as the technology evolves. Fluorometers were something that when I started my career, they were pretty few and far between and relatively expensive to use. And now, you know, we have the handheld fluorometers that we're using all over the place. So there's a lot of potential here to gain new knowledge, new understanding, and to better monitor our environment. And I'll conclude there.